The overall goal of this procedure is to identify protein binding regions via chromatin. This is accomplished by first cross-linking the protein to the DNA. The second step is to shear the DNA. Next, the DNA-bound target protein is immunoprecipitated and then eluted. The final step is to reverse the cross-link and to isolate the DNA. Ultimately, chromatin immunoprecipitation is used to show localization of protein binding across the genome.
This method can help answer key questions in the functional genomics field, such as identifying and mapping protein-DNA interactions in a given tissue or cell line.
The cells for this procedure are grown in 100 by 20 millimeter cell culture dishes. The amount of cells can range from 1 to 10 million cells per dish, depending on cell type. Approximately 2 million cells are sufficient for one immunoprecipitation. To cross-link protein and chromatin in the cells, add formaldehyde to 1% and incubate for 10 minutes at room temperature with occasional rocking. Quench cross-linking by adding glycine to a final concentration of 125 millimolar and incubating for 5 minutes at room temperature. Wash cells twice with 1x phosphate buffered saline. After the second wash, decant PBS and add 0.2 milliliters of fresh PBS. Harvest cells with a plastic cell scraper into a microcenter.